Hello guys, welcome to Big Scholars Academy. And today I'm going to teach you about the asexual reproduction. Principally, asexual reproduction is divided into 5 major kinds. The first one is fission, second one is fragmentation, third one is budding, and the fourth one is spore formation, and the fifth one is vegetative propagation. First, we'll learn about the fission. Fission. Basically, fission is divided into 2 major categories, that is binary fission and multiple fission. What is binary fission? Binary fission is something like when 2 individuals are formed from a single parent, then that's called as binary fission. The main example is bacteria, amoeba, leishmania, and etc. For example, there is a cell and the nucleus starts dividing in the first step. What's genetic material in it? The DNA. That means the DNA starts replicating. In the second step, cytoplasm starts dividing. A very important point, where nucleus starts dividing, that's called as karyokinesis. And when cytoplasm starts dividing, that process called cytokinesis. And in the third step, finally, the 2 daughter cells are formed and the cell wall is synthesized. And the another very important point is, for example, before dividing, the cell has 23 pairs of chromosomes, and after dividing also, the daughter cells will have 23 pairs of chromosomes. But whereas in multiple fission, where many individuals are formed from a single parent, then that's called as multiple fission. The very important example is Plasmodium, which causes malaria. And the process for this multiple fission is the first step. A protective layer is formed around the cell, like this. And now this cell is called as a cyst. Now, the cyst, the nucleus inside the cyst starts dividing many times. And after that, even the cell cytoplasm divides many times, and after that, the third step, the cyst starts breaking and by forming many individuals, and this is called as multiple fission. Fragmentation. Fragmentation generally occurs in colonies. It also generally occurs in multicellular organisms. So basically, what is fragmentation? Fragmentation is something like when a matured organism splits into many fragments and grows into matured organisms is called as fragmentation. The basic examples of fragmentation are cyanobacteria, flatworms, annelids, spirogyra, molds, lichens, sponges, etc. So the process of fragmentation is as follows. So in Hydra, how the fragmentation occurs. For example, this is a matured Hydra. So when it is cut into pieces, when it is cut into 2 pieces, for example, after few days or few hours, the remaining part grows. For example, a cell is divided, it's broken into 2 pieces. After some time, it grows into 2 daughter cells. So in Hydra, this is a process of fragmentation. But whereas in planaria, it's the same process. When a mature planaria is cut into 2 pieces, after few hours, a half day, the remaining part of the planaria grows. In spirogyra, when this spirogyra is cut into 2 pieces, after a few days, the remaining part grows. So this is called as fragmentation. Budding. A bud grows into a fully matured individual and eventually breaks away from the parent. The basic example of this is yeast. And I'll tell you how this process takes place. For example, this is a matured cell. For example, this is an yeast. So in the second step, a small bud occurs on the cell wall, and also the nucleus expands, and the bud enlarges, and even the nucleus starts dividing. And in the last step, the bud separates from the parent cell. So what is the difference between binary fission and budding? Seems to be interesting, right? So what in binary fission, what happens is when a single parent cell divides into two cells, they are exact like clones and of equal size. But whereas in budding, the daughter cell is a bit small than the parent cell. So this is the key difference between budding and binary fission. Spore formation. Many individuals are formed through spores. The main example of this is bread mold. Etc. So I'll say the process for this. For example, this is a bread mold. For in the first step, if you want to see this in real, take a piece of bread, wet the bread by sprinkling some water on it, and leave it aside for around 2 to 3 days. And after that, take a small piece and observe this under a microscope. You'll find something like this, and this is called as a bread mold. So it has many structures, and this whole bulb-like structure which contains spores in it. These are spores, and this whole structure is called as sporangium. Sporangium. And these root-like structures are called as hyphae. Now, how the reproduction takes place on a bread. So first, there is a bread mold. So what happens in the first step is the sporangium surface breaks, and a few spores gets out, and they form a small bulb-like structure containing a few spores. Yeah, as these spores are motile, they are called as zoospores. These spores travel some distance and they settle on some other surface of a bread. When they just settle down, some spores gets out of this bulb-like structure, and another bread mold starts growing from the same place. And again it releases some spores, the zoospores. It settles in some other part. Again another bread mold starts growing, and this reproduction takes place very fast, and this is spore formation. And guys, don't worry, I'll keep vegetative propagation as in another video as it's too large. But for today, this is the end.